The origin of species. In 1928, a German zoologist by the name of Ernst Mayr traveled to New Guinea to find samples of plants and animals for his work. Ernst Mayr is a particular lover of birds. He was very interested in birds, and one thing he noticed in New Guinea is that there were certain organisms, or most of the organisms, were all clustered into this one section. He was able to discover 137 species of birds, and noticed that these birds were all in these separate, discrete places, in what he defined as an organism space. He noticed that these clusters in all organisms they all reproduce sexually, which it means that these clusters are what he now defines as what you call species. With the species of birds he found in New Guinea, he was able to show the importance of speciation and through the concepts of the morphological species and especially the concept of the biological species. So first of all, how would you define a species? So one of the concepts we found is using the morphological species, which basically says which group of individuals that close resemble each other, meaning you would see two different species that look almost exactly alike. However, they could be two separate species. For example, we have the eastern and western meadowlarks. Notice that these two species look very similar to each other. They both have yellow bodies with the same type of color patterns. But one thing that separates them is that these meadowlarks have different songs, which defines them as each their own species. The morphological species is also very useful in helping in determining fossil species. By using the morphological features of the fossils, we can basically categorize them what type of species were they when the fossils was still alive back in the day. However, there are many different problems with the morphological species concept. First of all, it is not very easily testable. The morphological species uses the appearance of each organisms, and appearance doesn't always show everything. Like for example, the meadowlarks in the previous slide, they are very similar in morphological species, but they cannot interbreed with each other since they use very different songs. Such species like these meadowlarks are considered cryptid species because they're morphologically similar but very different in genetic makeup. Conversely, we have another problem with morphological species concept. We can have organisms that would have very different morphological features but have very similar genetic makeup structures. A prime example for that, as stated by Coyne, would be us Homo sapiens, the human beings. Like if you take two examples such as the Inuit people of Canada and compare it to the Kung tribe in Africa. These two groups of people have very different morphological features, but they're still both the same species. They are both human beings, hence they can interbreed each other. Sexual dimorphism is another problem for morphological species. It is often confused that the males or females are often different species because of their different patterns and colors. Often the case with sexual dimorphism, they often been classified as two different species, but in fact they are one distinct species.
Biological species concept. It is considered to be the golden standard for evolutionary biology. It was proposed by Ernest Mayr and Theodosius Dobzhansky. The definition is a group of interbreeding natural populations that are reproductively isolated from other such groups. Reproductively isolated refers to members of different species have traits that differentiate one another that make them unable to interbreed. These traits can be through appearance, behavior, and physiology. An example of a reproductively isolated organism would be a mimulus monkey flower, because there are two different types that can be found in the Sierra Nevada. They both live in the same environment, but they have different pollinators. One flower over here is being pollinated by a bumblebee, and another is by a hummingbird. E- Each flower is specific to a specific pollinator. For even though they are known as a mimulus monkey flower, each of the flower has changed its ways to accommodate to the certain pollinator it wants. So, therefore, it cannot simply interbreed if a hummingbird goes to a bumblebee's mimulus monkey flower or vice versa. An important thing to consider is that species is considered a reproductive community. So, in a reproductive community, as each generation of the organisms continue, mutations will end up occurring. If the mutations were to be good, then the certain mutation would stay within that species and not transfer over the course of interbreeding to a different but similar organism. This makes them their own unique way of differentiating themselves from the general species. The lack of interbreeding with other species helps them maintain the species' difference in trait. When interbreeding does occur, most of the organisms that have made it are unable to develop and therefore have zero fitness. But there are rare cases where there is interbreeding within a group of species that create a live offspring. For example, the mule. A female horse and a male donkey mate and make a sterile mule. But unfortunately, this hybrid is unable to exchange genes with another mule. So, therefore, the hybrid cannot continue in its reproductive state.
The final prediction on geographic isolation states that the reproductive isolation between a pair of physically isolated populations increases slowly with time. The perfect example of this specific prediction would be fruit flies or Drosophila species. The author of this book, Jerry A. Coyne, and his colleague Alan Orr, studied pairs of fruit fly species that diverged from a common ancestor long ago. Coyne and Orr found that reproductive segregation between organisms increased dramatically with time, and with this, leading to completely preventing the hybrids to interbreed after they diverged about 2.7 million years ago. And although it is a slow process, divergence could form new species in this manner. Speciation commonly starts off when reproductive isolation is absent. Then isolated populations diverge. When the divergence has gone for a sufficiently long time, reproductive barriers develop, which then leads to complete speciation. When new species arise without the need of geographic isolation, it leads to sympatric speciation. An example is the species of cichlid fish found in the lakes of Cameroon in Central Africa. There are two small lakes wherein cichlid fish organisms habituate in. But one lake has 11 species and the other has 9. Another example is the Kentia and curly palms in Lord Howe Island off the coast of Australia. The palms appear to have diverged from an ancestral palm that lived about 5 million years ago. These are sympatric speciation because the habitats are too small to contain any geographic barriers. Sympatric speciation is very rare despite given the opportunity. Coyne and another colleague of his, Trevor Price, surveyed many bird species of 46 isolated oceanic islands, and none were each other's closest relatives. Despite being rare, sympatric speciation is very common among plants due to polyploidy, a mechanism that changes chromosomal numbers. There are two types of this mechanism. Allopolyploid speciation is when two different species in the same area create a hybrid, and this hybridization process basically doubles the chromosome number and produces fertile hybrids that would then give rise to new species. Autopolyploid speciation is the doubling of chromosomes and production of fertile hybrids from self-fertilization. Polyploidy is very recurrent among flowering plants because most flowering plants self-fertilize. Apart from self-fertilization though, Welsh groundsel or Senecio cambrensis is a good example of allopolyploidy and is proven to be a polyploid hybrid of common groundsel, Senecio vulgaris, and Oxford ragwort, Senecio squalidus. Polyploidy is rare in animals because they are unable to self-fertilize. Overall, polyploidy is a fast process because new species could arise in just 2 generations. So where did species originate from? Surely from a common ancestor. And with the help of two main players, geographic barriers, and reproductive isolation, speciation occurs.